medicine. I graduated in 2011 from the University of Wisconsin with a degree in medical microbiology and immunology. And I'll be starting here in August with the med school in 2014, so this year.
How did you get involved in research?
I started as a student in Biology 152. It was a course that I took during my undergrad here, and one of the things that you could do would be a mentored research project. And so I started working for Dr. Timothy Camp and did my research project with him, stayed with him for about 2.5 years, and then wasn't quite ready to go to med school, so I was looking for jobs. And Dr. Ravi Baljapali, who is my current PI, was Dr. Camp's postdoc, and they work pretty closely together, collaborate on a lot of projects. So I started working for him, and I've been with him for about 3 years. And
so tell us about your research.
So this LRC10, leucine-rich repeat-containing 10 protein, is what that stands for. It's a protein that hasn't really been shown in any humans quite yet, except for just recently Mayo Clinic has found an infant with dilated cardiomyopathy with a mutation in this protein. So it's becoming an exciting field, and hopefully we'll be able to make some correlations here. But for our purposes, we're just testing some mice that had a mutation in LRC10, and what we were doing was we were measuring calcium currents. So taking a cardiomyocyte, a single cell isolated from heart, and passing an electrical current through it, and seeing whether or not the currents would be normal compared to a normal phenotype to these mice that have dilated cardiomyopathy phenotype. So what we found in these mice is that this calcium current, L-type calcium channel, one of your main currents that is involved in your action potential that generates your heartbeat, was severely diminished in these mice compared to their wild-type littermates. So that kind of got us interested, and we started looking at this protein and found that it does, the protein LRC10 and CAV1.2, which is your L-type calcium channel, do associate with one another on the protein level. So we're just starting this. If you do a PubMed search, there's about 6 papers that show up with LRC10. So it's a pretty novel protein, but now that there is a human case, it's getting everyone pretty excited, at least in our lab and the collaborators at Mayo Clinic, that it might become something, and possibly what we're kind of thinking is that LRC10 is possibly an auxiliary subunit for L-type calcium channel. And the L-type calcium channel has been studied for years. There's a lot known about it, and so this protein having a correlation is kind of a new thing that we're getting kind of excited over.